Okay, so let's look at the first. Okay, for the first part of macromolecules, which is part A, I'm going to give you the answer. Okay, the answer, and then you check with your answer. Is it correct or not? If you have question, you can ask. All right. So for question number 1, the answer will be B. Okay. Question number 2 is A. Number 3, D. Number 4, C. Number 5, D. Number 6, A. Number 7, D. Number 8, C. Number 9, A. Okay, number 10, A. Number 11, C. Right? 12, D. 13, B. 14, A. 15, B. 16, B. 17, A. 17. Okay, 17, A. 18, C. 19, B. 20 is C. 21, B. 22, B. 23, D. Okay, 23, D. 24, C. 25, C. Okay, so for all the MCQ questions, do you have any question related to part A that you don't understand that you want me to elaborate or explain? Ada soalan?
Tak ada, madam.
Okay. Okay, so moving on to the next part, all right? So let's look at the next part, part B. Okay, so part B, question A. So it shows you the interactions between water molecules. So obviously, water molecule will be attracted to each other through hydrogen bonds. Okay, so the answer for A is hydrogen bonds. Okay, so and then next question, explain 3 properties of water that can be related to the bond in A. Okay, so the answer for A is hydrogen bonds. Okay, so for this question, for question number 2, you of all the 7 properties of water, so you have your notes. In your notes, there's a slide showing there's 7 properties of water. So of that 7 properties of water, you have to choose 3, and you have to explain how these 3 properties of water relates with the formation of hydrogen bonds between the water molecule. Okay, so for the first properties, you can answer. For example, water is a good solvent. Okay, so why is it a good solvent or universal solvent? Because it can form hydrogen bonds with polar molecules such as glucose, or with ions. Okay, so ions such as we have, for example, in your notes, there's ions that form between sodium ion and chloride ion, which is salt. Okay, salt, when it dissolves in water, it will be surrounded by water molecules, okay, to form hydration shells. Okay, so the water that surrounds this ion, it causes the salt to be dissolved in water through the formation of hydrogen bonds. So water is basically a good solvent because it can attract ion and also polar molecules through hydrogen bonds and causing this molecule to dissolve in water. Okay, so the second properties of water is that it has high surface tension. Okay, high surface tension or adhesion, where the water can stick to certain to other substances that are also polar. Okay, it has high surface tension due to hydrogen bonding between water molecules and other molecules. Okay, so that is for surface tension. Okay, surface tension. If you look in your notes, Okay, you have, for example, on the surface of water. So the water molecule at the surface, it forms hydrogen bond to one another and hydrogen bonds to the water molecule below. That forms surface tension. So this allows, for example, insects to be able to walk on the surface of the water. So kalau kita nampak dekat sungai ke apa kan, serangga boleh berjalan atas air tu. It is due to surface tension, okay, or adhesion, water sticking to other molecules that are also polar, okay. And then the third properties that you can include in your answer is that it has high specific heat capacity. Okay, high specific heat capacity. So this high specific heat capacity, kenapa air tu perlu serap kuantiti haba yang sangat banyak? Sebab this is due to the hydrogen bonding between the water molecule are so much in order to increase the water molecules temperature. Sorry, the water temperature. So water needs to absorb really high amount of heat. Okay, this is, for example, to vibrate the water so that it leads to the next property, which is water has high heat of vaporization. Okay, high heat of vaporization, dia sinambungan daripada high specific heat capacity lah tu tadi kan. So heat of vaporization is the heat a liquid must absorb for 1 gram to be converted into gas. So air tu perlu, air dalam bentuk liquid, it has to absorb so much heat so that the hydrogen bonds, especially hydrogen bonds at the surface of the water, can be broken so that the water that is in liquid form can be released as gas, as gas. So the hydrogen bonding individually it is weak, but when there's so much of the hydrogen bonds form, the water basically needs quite a large amount of heat to break these hydrogen bonds between the water molecule. And then the other properties that you can include is that obviously water in solid form, it expands. Yeah, so basically water will have maximum density at 4 degrees Celsius. If fall below 4 degrees Celsius, the water molecules will expand in order for it to form a crystal ice structure. Okay, so therefore water in solid state, which is ice, will be less dense compared to water in liquid form. So this is due also to the formation of hydrogen bond. So hydrogen bond between water molecule in solid state, okay, is fixed. So one water molecule form hydrogen bonds with another water molecule, and the formation is fixed so that it can form ice crystal structure. If you refer to your notes lah kan. So ice will be less dense compared to water in liquid form. So maximum density of water is at 4 degrees Celsius. So if you look at these questions, actually, okay, so this is how you answer question dengan cara yang betul. Saya nak cerita sikit. Okay, soalan ni disuruh bagi 3 properties. If in your answer you just give, okay, water is a good solvent, water has high specific heat capacity, water has high heat of vaporization or high surface tension, without explaining, then your answer will be wrong. Dia tak ada setengah-setengah markah ya dalam exam, especially untuk bio. Bio kita ada setengah-setengah markah. Dia markah 1 full. So when the question asks for explanation, you have to explain. So explain lah tiga properties and relate how this property related to the formation of hydrogen bonds between water molecule. Okay. So next question, explain the property of water when it changes from liquid to ice. Okay, from liquid to ice. So tadi basically saya dah explain lah. So water expand upon freezing, the first point. Okay, water expands upon freezing, or it is less dense. Ice is less dense, or it has lower density compared to water in liquid form. Why? Because due to the many hydrogen bonds formed between water molecules. Okay, so for the third point, one water molecule bonded to 4 other water molecules, and this is to form, okay, form strong rigid hexagonal crystal structure. Okay, so the hydrogen bonds between the water molecule will be more stable. Alright. If you have question, you can ask. So saya bagi je jawapan. Kalau saya bagi jawapan and then
yang untuk tadi tu apa? One water molecule bonded with 4 water molecule untuk apa? Form apa?
Rigid, okay, strong rigid hexagonal crystal structure, ice crystal lattice structure tu kan?
Madam dia tolong eja tau Aidon.
Yang mana?
Yang tu, yang panjang-panjang tu.
Rigid, strong rigid.
RIGID.
Dia fix maksud dia. Ikatan hydrogen between water molecule is fixed. Rigid means it is fixed. It cannot be easily broken. Macam tu. It is rigid to form hexagonal crystal structure, ice crystal structure tu. Bila dia form ice crystal structure between water molecule tu, jarak antara air tu akan lebih jarak lah, faham tak? Berbanding dengan kalau in liquid form, dia lebih rapat. Sebab apa? Water molecule will form hydrogen bond and then breaks and then reform. So dia boleh bergerak, dia boleh dekat, dia boleh jauh kalau dalam bentuk liquid lah. Tapi kalau dalam bentuk ice, it is fixed. The formation of hydrogen bond between water molecules are fixed. Okay? Boleh?
Boleh.
Okay. Hexagon tu hexagon lah, bentuk hexagon. Okay, crystal, ice crystal structure pun boleh. Ice crystal structure. Okay, so next question. Figure 7 shows. Any question?
So basically kita explain about the height density lah kan?
The density and then the formation of hydrogen bonds between water molecule are fixed. One water molecule form 4 hydrogen bonds with another 4 water molecule. Hydrogen bond tu mesti awak kena explain lah. Kan kalau dalam bentuk ice, ikatan hidrogen antara water molecule tu memang fix. Satu dengan 4 lagi yang lain. Macam tu. Okay. Okay, sekejap saya macam nak kena conteng dekat saya punya screen ni. Cumanya saya tak setting dia lagi. Okay, so while waiting, so if you look at this diagram, okay, figure 7 shows water molecule forming hydration shells, okay, around ions of copper 2 sulfate, okay. So basically copper, CU copper kan, okay. Okay, so copper, the charge is 2 plus. Boleh nampak saya conteng ni? SO.
Boleh.
Okay. To negative, okay. So we have here the cation and we have here the anion. Okay, so in order for this substance or molecule to be dissolved in water molecule has to surround these ions. Okay, so when water molecule surrounds this ion, we call it as hydration shell. Inilah kan hydration shell. Ini lagi hydration shell, kan hydration shell. So obviously, if you look at the formation of hydration shell surrounding these ions, so molecule P, the region of water molecule that faces ion P, it is the hydrogen ion region. Okay, the hydrogen atom region of water molecule. So hydrogen it has partial positive charge. So obviously P is SO sulfate ion kan. So R it has oxygen region facing the R ion. So oxygen has partial negative charge. So obviously R is Cu2+. Okay. So saya harap awak faham ya konsep hydration shell ni. Apa-apa yang menyebabkan sesuatu substance tu larut dalam air, sebab air forms hydration shell. Water molecule surrounds those ions or polar molecules. Okay. Okay, so next questions regarding again on basically bila kita tengok water strider ni surface tension lah kan. So water strider which is an insect that can glide and hop on the surface of water without sinking. So explain how the properties of water enables it to do this. It is due to what? The attraction between water molecules. So the attraction between water molecule, we call it cohesion. Okay, cohesive behavior. Okay, cohesive behavior between water molecule. That is the first point. Okay, first point. So cohesive behavior between water molecules. First point. Second, causes the water to have a high surface tension. Okay, it causes the water to have a high surface tension. Yang ketiga, I will add another point. Okay, so surface tension is due to what? Due to hydrogen bonding between the water molecules. The formation of hydrogen bonds between water molecule occurs where? Between water molecule at the surface, okay, and water molecule below. So for example, here we have a lake. So ini saya dah draw dah kan kat dalam kelas, sama je. So we have water molecule at the surface, okay, of a lake, and then water molecule below, okay. So the attraction between water molecule is through formation of hydrogen bond. So this is water molecule at the surface. We call this as air-water interface. Okay, air-water interface. The formation of hydrogen bonds between water molecule at the air-water interface and also the water molecule below. This is below. Okay, below. So this forms surface tension and allows an insect to be able to walk on the surface of water. So saya bagi you all ada 4 point kat situ. Okay, 4 point. I repeat, cohesive behavior, water has high surface tension due to hydrogen bonding between water molecule at the air-water interface and the water below. Okay, so ada 4 point, dia nak 2 markah saja. So kalau cakap cohesive behavior dah dapat markah. High surface tension pun dah dapat markah. Macam tu kan, right? Tapi kena explain lah, you have to explain, you cannot put in point form. Okay, you have to explain. When you are answering questions, especially in exam, you have to answer in full sentences. Okay, not in point form as in your notes. Okay, your own notes lah basically. Kita bila buat nota belajar kan kita buat short form lah. That you don't apply during exam. Okay, so C. In plants, water absorbed is transported through the xylem. How do adhesion and also cohesion help in the movement of water from root to shoot? So this question is 3 marks. So basically, soalan macam ni dia sebenarnya kalau dia adalah soalan macam applied lah, it gives you a situation and then you have to explain. Okay, so this question it is worth 3 marks. So basically, adhesion and cohesion. Adhesion is water sticking to the cell wall of xylem, kan? And cohesion is water sticking to each other to form a column of water, right? So the first point should be it involves cohesion, okay? Cohesion, which is water molecule attracted to one another, okay, through hydrogen bonds, okay, to form a column of water, okay, a column of water within the xylem. Okay, second point, okay, you have adhesion, okay, so adhesion. So adhesion is when water sticks, okay, water molecule sticks or attracted to cell wall of the xylem. Alright, number 3, disebabkan oleh apa? Due to hydrogen bonds, okay, due to hydrogen bonding, okay, hydrogen bonds. So the next point you may add, for example, extra point. Dia tak ada extra point, maksudnya you can elaborate more, okay. So cohesion helps pull together the column of water in the xylem, kan. So kita ada xylem and then air, kan. the water molecule will be attracted to one another to form a column of water. And in order, so this is cohesion. And in order to prevent the water molecule from falling back down due to the pull of gravity, okay, so the water molecule will be sticking to the cell wall of the xylem. So this one is adhesion. Okay, adhesion. So adhesion help resist the downward pull of gravity. Okay, it helps resist the downward pull of gravity. So cohesion and adhesion, okay, create on the column of water. So water can be transported from roots, okay, in the soil, until it can be used to carry out photosynthesis in the leaves, for example. Okay. So photosynthesis kita tengok nantilah chapter 6. Okay, the detail of photosynthesis process. Okay, so the last question for number 1, question 1 ni. So in the table below, explain the arrangement of water molecule and hydrogen bonds in different states of water. Okay, so in vapor, ini daripada sekolah lagi kan. So for this question, you look at the question carefully. Dia nak suruh awak explain apa? Susunan air, kan, the arrangement of water and how it relates with hydrogen bonds. Yeah, so susunan tu kena relatekan dengan hydrogen bond lah. So the water molecules in vapor state or gaseous state will be further apart. The water molecules are free to move. Okay, so therefore the bonds between the hydrogen bonds between water molecule are easily broken. They have loose hydrogen bonds. Okay, they have loose hydrogen bonds that breaks easily between water molecules. Hydrogen bonds, okay, that breaks easily. Between water molecules. Water molecules are more free, can they are freely move further apart from each other. Okay. So for ice, okay, the formation of hydrogen bonds between the water molecule are more fixed. It is more rigid. Okay, more rigid. It has strong and stable hydrogen bonds. Apa boleh? Strong stable hydrogen bonds. Okay. Strong or and pun boleh. Strong and stable hydrogen. Hydrogen bonds between water molecules that forms large gaps. Okay, it forms large gaps between the water molecule, causing ice to be less dense. Okay, less dense, right? Ataupun for the same point, I boleh explain juga each water molecule can form 4 hydrogen bonds with 4 other water molecules. Okay, 4 hydrogen bonds with 4 other water molecules. Right? Ataupun point banyak je point boleh letak kan. So ataupun boleh letak hydrogen bonds between water molecule forms. It forms crystalline lattice structure. Okay, dan tadilah sama je, crystalline lattice structure. Okay, structure. Okay, so this question total dia semua 15 markah lah. Alright, untuk soalan ni lah for this question. Alright, okay, so moving on so far for question number 1, do you have any question? Saya tak boleh read you all punya comment sebab saya buka saya punya screen ni. So if you have question, just turn on your mic, then we can discuss further lah. Jangan komen, saya tak nampak. You have to switch on your mic. Okay. Okay, so next question, figure 8. Kita akan cuba jawab soalan ni, habiskan exercise chapter 1 ni malam ni. Alright. So question 2, figure 8 shows the structure of an amino acid serine. Okay, so amino acid we have 20 different types. Okay, one of it is serine. Okay, so in this question it is serine. So state the variable group. So the variable group is also the as the R group or the side chain lah, sama je. Okay, side chain. Okay, so variable group which is this one. Okay, tulislah CH2OH. Okay. Circle dia tak dapat markah tau sebab dia suruh state. Okay, our circle kat sini, your answer will be wrong because you are not answering what the question wants. The question wants you to write, okay, the side chain. Okay, so figure 9 shows the molecular structure of an amino acid. Draw the zwitter ionic form of an amino acid. So amino acid usually are found in your cells, in living cells. So living cells are aqueous. Again, it has water inside. So it causes the amino acid to be able to ionize in aqueous surrounding. So when amino acids ionize in the aqueous surrounding, okay, so the carboxyl group here will lose the hydrogen atom. Okay, so carboxyl group basically it becomes proton donor lah. Okay, proton donor. So it releases hydrogen ion into the surrounding and accepted by which functional group? It is accepted by the amino group. Okay, so because carboxyl group loses a proton, so it will be negatively charged. Because amino group receives the proton, it will be positively charged. So the amino group here it function as proton receiver and proton acceptor.
Miss, madam. Untuk variable group tu, jawapan dia boleh jadi R group ke?
Tak boleh sebab R group tu dia basic formula of an amino acid. If a question gives you a diagram, okay, if a question gives you a diagram, so it means that the diagram gives you information. Okay, so you have to use the information given in the diagram. So soalan 2 ni, ini kan dia bagi diagram 8 ni. So you have to refer to the diagram. Same goes to question 9. So question 9 ni kan. So lah for example R group ni dia bagi spesifik CH2 lah dia bagi kan. Sorry CH3. 1 2 3 CH3. Okay, CH3. So kalau soalan macam ni dia bagi spesifik structure of an amino acid kan, jadi dia R, walaupun dah buat ion ni betul kan, ada positive charge dekat H, ada negative charge, but because you don't use the diagram dengan betul kan, apa balai tukar, jawapan awak akan boleh macam tu. Okay, so you must use the information given in the diagram. So in this case, draw lah balik. Jangan draw dekat sini tau. Draw balik. So bila dia bagi R kat sini, awak tulislah R. Tapi kalau dia bagi another atoms, a group of atoms, you draw exactly what the diagram gives you. Okay, so positive. So this is the zwitter ionic form. Okay. Okay, so amino acids, boleh tadi? Okay, so next question, it gives. Okay, so next question, amino acids are amphoteric molecules. So I explain the importance of amphoteric properties. So amphoteric, okay, what is amphoteric? Basically, it is a molecule that can function as both base and also acid. So that's why the name is amino acid. It can function as both base, okay, amino group, and also acid due to the presence of carboxyl group. Okay, so explain the importance of amphoteric properties. The first point, it has both basic and acidic group. First point, okay, or the amino group. Okay, this amino group is ionized to become NH3+, and mesti ada N. The acidic group is ionized to become COO negative. So what is the function or benefit of being both base and also an acid, it can function as, second point, pH buffer. Okay, pH buffer. And then third point, more elaboration, which is to maintain, okay, maintain pH of a solution, okay, maintain pH of a solution ataupun resist changes in pH of a solution. Dia akan menyebabkan pH dalam sesuatu solution tu stabil. Contohnya kita punya darah, kan, darah kita, our body fluids, especially your blood, it has to be maintained at pH 7.4. How to maintain this pH? Your blood will have so many different protein, especially hemoglobin. Okay, hemoglobin later next hemoglobin ni sebenarnya dia boleh bind kepada hydrogen ions. Okay, hydrogen ion. So macam contohnya bila awak exercise dengan kadar yang banyak, awak exercise kan petang-petang, pergi jogging ke main bola ke whatever, obviously you produce so much carbon dioxide, okay, so much carbon dioxide. So when you are respiring, your body is respiring, your cell is respiring, the cells produces so much carbon dioxide, and this carbon dioxide will make your blood to be acidic sebab dia akan form carbonic acid. Okay, carbonic acid. So the formation of carbonic acid causes the release of hydrogen ions into your body fluid, into your blood, and then this hydrogen ion will bind to the hemoglobin. Hemoglobin, we know that it is protein, kan? It is protein. So this excess release of hydrogen ion into your blood will bind to the hemoglobin. Okay, hemoglobin is protein, function as pH buffer to maintain your blood pH at 7.4. So 7.4 is the normal pH of your blood. Okay, body fluid. Okay, so give an example of a defense protein, antibody, immunoglobulin ataupun short form dia IG. Okay, bukan Instagram immunoglobulins ataupun lysozyme. Okay, lysozyme, inside your cell there's an organelle, we call it as lysosome. Okay, lysosome, inside lysosome there's an enzyme, we call it as lysozyme. Lysozyme tu macam just kita dalam awak punya sel ni ada white blood cell. Ya, dalam badan awak ni ada white blood cell. So white blood cell ni ada macam-macam jenis. Salah satu jenis dia adalah macrophage. Okay, macrophage. So macrophage ni fungsi dia kan, dia akan engulf pathogen. Okay, dia akan engulf pathogen. So when this macrophage engulf a pathogen, so this pathogen will be surrounded by a vesicle. Okay, so a vesicle. So inside this macrophage also there's the lysosome. So the lysosome that contains lysozyme, which is a hydrolytic enzyme, will fuse with the vesicle containing the macrophage, the bacteria, for example, or any type of pathogen, and the lysozyme degrade the pathogen inside into debris. Okay, so that's a process, we call it as phagocytosis. Okay, it involves lysozyme inside lysosome. Okay, lysozyme is enzymes. Enzyme is protein. Okay. Okay, so next question, figure 10 shows the structure of an amino acid isoleucine. So isoleucine is another type of amino acid where the side chain here it consists of many carbon and also hydrogen. So as you can see, because it has many carbon and hydrogen, we know that carbon and hydrogen they have the same electricity. So this is the side chain that is non-polar. Non-polar. So this is a type of non-polar amino acid. Okay, non-polar amino acid. So draw a circle to show the side chain of amino acid. Saya dapatlah kat situ circle lah kan. Tapi kalau dia nak circle, cantiklah circle awak tu. Okay, circle kan. Okay, circle cantik. Alright. So kadang-kadang kalau buat careless macam ni kan, tiba-tiba miss out hydrogen macam tu. Jadi salah So circle carefully to include all the atoms, okay, that you're supposed to circle. Okay, so number 2, explain what happens if isoleucine is dissolved in water. Okay. Tadi kan Tadi madam kata isoleucine ni non-polar. Tak patutnya tak boleh lah dissolve in water kan. Tapi soalan ni tiba-tiba dia cakap dissolve in water pula. Macam mana kan contohnya. Okay, basically non-polar amino acid, it gives hydrophobic properties for a protein. Okay, it gives a hydrophobic properties for a protein. So individually, if the isoleucine exists as a molecule like this, it can be ionized, maksud dia, can be ionized to form a Zwitter ion. Tapi kalau kita ada polypeptide chain macam ni, kan, polypeptide chains like this one, and then you have isoleucine here, you have isoleucine here also, you have isoleucine also here. So it makes this polypeptide chain to form a protein that is that has the property of hydrophobic, kan, sebab dia banyak sangat isoleucine, which is non-polar, kan. So it makes the protein later to have a hydrophobic property, non-polar property, does not attract water molecule. Kalau kita belajar nanti kan chapter 3, kita akan belajar, okay, dalam plasma membrane ni kita ada apa nama, phospholipid bilayer, right? Okay, phospholipid bilayer. Apart from having phospholipid bilayer embedded in between, you have proteins. Okay, proteins that function as transport protein, for example, that allows molecule to pass through. Okay, so this protein here, the region that is in this region here, the hydrophobic region, are made up of amino acids that are not it has hydrogen. It has both hydrophobic region and also hydrophilic region. So awak jangan ingat phospholipid molecule je ada hydrophilic and hydrophobic region. Protein embedded in the plasma membrane also have hydrophobic and hydrophilic region. Okay, so explain what happen if isoleucine dissolve in water. Okay, so tadi lah sama kan. So if it dissolve in water, it will be ionized. Kan, first point, it will be ionized or dissociate to form Zwitter ions. Tu satu point yang pertama. Okay, isoleucine dissociates to form Zwitter ion, or the molecule has positive and negative charge. Okay, positive and negative charge at the functional groups. Okay, so second point, positive and negative charge tu kat mana, kan? So at the amino group lah, kan? So amino group ataupun NH2, kan, will be ionized to form NH3+, kan. So the amino group is ionized to form NH3+, or it becomes a proton apa? Acceptor, kan? Acceptor. For the third point, the carboxyl group COOH will be ionized to form COO negative. So this becomes a proton donor. Okay, so yang first point tadi ionize lah, ionize or dissociate to form Zwitter ions. Okay, Zwitter ions. It has both positive and also negative charge, okay, at the functional groups. Ataupun it is a bipolar molecule. Okay. Jangan ingat manusia je bipolar, molekul pun bipolar juga. Okay. Okay, so draw a dipeptide, and label the peptide bond. Okay, so for this type of questions, okay, it does not give you any diagram to help you to draw a dipeptide. So that's why I said earlier, you have to remember and memorize the basic structure of an amino acid. Then only you can answer this type of question. Okay, so dipeptide basically consists of two amino acids bonded together by a peptide bond. So this question is 2 mark. Okay, so first you have to draw lah a molecule of an amino acid. Kita ada chiral carbon bonded to a side chain. So I'll draw just basic je. Basic structure of an amino acid. So hydrogen, on this side we have the group. Okay, on this side we have the carbonyl group. Okay, so carbonyl group during the formation of a dipeptide, which is from 2 amino acids, kita imagine kat sini kita ada OH, kan? kat sini, sekejap. Okay, kat sini kita ada OH. Okay, so another amino acids dekat sebelah dia. Okay, you draw the same thing, carbon and then R group and then H, carbonyl group and then the amino group. Okay, so in order for you to attach these two amino acid together, a water molecule will be removed, kan? So this removes a water molecule. So you padamlah balik. Padam balik, kan? So and then you just join the two atoms, which is carbon and also nitrogen together. So yang ni macam tu lah, kan? Imagine yang ini kita rapatkan lagi lah. This the formation of nitrogen peptide bond. Okay, peptide bond. So this mark this question is basically it is worth 2 marks. So markah yang pertama kat mana? Markah yang pertama, you draw the structure of amino acid correctly. So the correct structure of amino the correct structure of a dipeptide is marked. The second is bond. Ini jangan confuse ya. The molecule is dipeptide, the bond is still called as peptide bond. Jangan tiba-tiba label dia dipeptide bond pula. Salah peptide bond. Saya cakap macam ni kenapa? Sebab ada yang jawab macam tu. Peptide bond. Okay. So next question, question 11. Question E, figure 11 shows a dipeptide. Inilah molekul dia, cuma yang ini dia dah bagi specific side chain dia. Ada CH3. So basically these are the same amino acid attached together. Okay. So draw one of the amino acid and boleh je kan. So the process of breaking the bond between carbon and nitrogen is hydrolysis. Okay, ini revision lah. Hydro means that it requires a water molecule. Lysis is to break the bond, the peptide bond here. Okay, so when you break the bond between carbon and nitrogen, so you add atoms from the water molecule, kan? So the carboxyl group receives OH, the amino group receives the nitrogen. So the product will be senang je lah kan kat situ. C double bond O, dia receive OH daripada air. Ada yang lain tu copy balik lah. Okay, so this is the product. Okay, the product form. Let's say lah soalan macam ni dia dah bagi structure and dipeptide tu, the specific structure of the amino acid that you should draw. Tiba-tiba apa pandai tukar, dia jadi R kat sini. Salah. Faham? So that's why you have to use the information given in the questions. So soalan ni soalan E, anak dia 1 kan. So bila E anak dia 1, okay, ni lah refer kepada diagram daripada soalan ibu dia, asal dia. Okay. Okay, so number 2, identify the group of amino acid that E1 belongs to. Dia ada apa? CH3 kan. So it is non-polar amino acid. Okay, non-polar amino acid ataupun hydrophobic is also accepted. Okay. Okay, so next question. Figure 12 shows the tertiary structure of a protein. Identify A and B. Tertiary structure, kan? It is for globular protein. Sorry. Okay, globular protein. So globular protein is the kita ada 2 protein based on structure. Kita ada satu fibrous protein kan. Fibrous protein, kita ada globular protein, the next one. So fibrous protein does not attain or does not have tertiary structure. Only the globular protein have the tertiary structure. So globular protein basically are protein that has metabolic function. Okay, it has metabolic function. Contohnya apa? Contohnya enzyme kan, enzyme it involves in as catalyst kan, as catalyst to catalyze reaction inside your cells or inside living cells. Ataupun kita ada hormones kan, hormones are another example of globular protein. So hormone it regulates your body function, okay, to properly. For example, if you eat too much sugar, too much rice, too much carbohydrates, your blood sugar level will increase. How to decrease back to the normal set point? Your pancreas releases insulin. Insulin is a type of hormone made up of protein. So you bring back down your glucose level back to the normal level by converting excess glucose into glycogen, for example. Okay. So another example of globular protein we have tadi apa? Antibody kan, antibody. We have hemoglobins. So this globular protein will have metabolic function and it can dissolve in water, and all the globular protein must have tertiary structure. Obviously kalau fibrous ataupun globular, dia akan ada primary structure, obviously kan. Primary amino acids are bonded together through peptide bond, and then they also have secondary structure, secondary helical ataupun pleated kan, helical ataupun pleated. So fibrous and globular kena ada primary, kena ada secondary. Cuma kalau fibrous dia akan skip tertiary, globular has tertiary structure. Okay, so kalau quaternary boleh ada pada mana-mana, globular ataupun fibrous, depending on the type of protein. Okay, so for tertiary structure, kenapa pentingnya globular protein ni ada tertiary structure? It is to allow the polypeptide chain to fold into specific shape. Okay, so the protein folds into specific shape to allow the protein to be able to function properly. For example, enzyme kan, enzyme kena ada active site kan. So active site ni disebabkan oleh protein tu fold into specific shape yang menyebabkan dia boleh ada structure yang ada active site for substrate to bind. Okay, so the tertiary structure is due to what? The interaction between side chains of amino acid along a polypeptide chain. So yang kelabu ni sebenarnya dia adalah backbone of a polypeptide. Backbone tu maksudnya apa tadi? Backbone tu adalah repeating unit of nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, all the way through. So we have here, for example, one amino acids, kan? Terbanyak amino acid so kat sini ada, kan? So how does tertiary structure achieved? It is due to the interaction between the side chain of amino acids along the polypeptide chains. So kat sini contohnya kita ada amino acids yang ada hydroxyl. And on this side here we have amino acid that also has a carbonyl group. Okay, so between hydrogen and also oxygen, interaction will form a hydrogen bonds. It causes the polypeptide chain to be folded into a specific structure. Okay, and then this region kita ada amino acid dengan side chain yang non-polar. Kat sini pula ada amino acid yang non-polar juga, with big interaction. Contohnya, kalau on this side pula amino acids yang ada sulfhydryl group SH kan, so it forms disulfide bridge. Okay, ini antara amino acids yang ada carboxyl group and also amino group kan. So carboxyl will be ionized to form COO negative, it is negatively charged. Amino group will be ionized to form NH3+, positively charged. Positive and negative forms hydrogen ions. Sorry, hydrogen bonds. Okay, bukan hydrogen bond, ionic bonds. Okay, ionic bond. This one is hydrogen bond. Okay, so A is hydrogen bond, B is ionic bond. Yang lain-lain tu pun mesti awak kena tahu juga. Okay, yang saya dah explain dah tadi. Non-polar, hydrophobic and also van der Waals kat sini, van der Waals interaction. Okay, hydrophobic and van der Waals. Okay, kita habiskan soalan ni kejap. Soalan Soalan on protein ni. Okay, so protein can be classified according to their function. Give 2 examples of protein with their respective function. Soalan ni pun sama juga. Give 2 example with their respective function. So kalau awak bagi enzyme je, awak bagi hormon salah. So it means that you have to explain what is the function of the enzyme, what is the function of the hormones. Kan? So protein, if you classify protein based on function, ada banyak. Kan? So ada protein yang boleh bertindak sebagai enzyme, ada yang bertindak sebagai hormones, kan? But you have to explain. Enzyme to catalyze chemical reaction inside cell. Hormone is to coordinate body function. Dan kita ada transport protein to move substance across cells. Kita ada antibody, kan? Antibody as defense proteins to attack pathogen, for example. Okay, and then kita ada albumin as storage protein in eggs to nourish embryo, contohnya, kan? So ada banyak lah. And then kita ada apa lagi? Structural proteins provide structural support. Okay, kita ada motor protein, function in cell movement, actin, myosin. Kita ada receptor proteins. Okay, receptor proteins to bind to signaling molecules such as hormones. Okay, out of the cell. So ada banyak, ada dalam otak awak, ada dalam jadual, kan? So just ambil lah 2. So hafalkan. Okay, so figure 13 is a fibrous protein. Okay, so fibrous protein, tadi kita tengok globular, so this one is fibrous. So feature or properties of fibrous protein, it is insoluble in water. Okay, it is insoluble in water. So draw the basic monomer of X. X is basically a polypeptide chain lah. So basically disuruh you all lukis amino acid. Okay, and name one of its functional group. Functional group kita ada apa? Amino group and also carboxyl group. Tu je. Okay, so draw the basic structure of an amino acid. Saya tak nak draw dah. Saya dah draw banyak kali tadi, kan? So draw the structure of an amino acid. So functional group dia amino group, carboxyl group. Okay. So next question, describe the level of organization of protein. So kita ada berapa? Kan, kita ada 2. If proteins combine that involves more than one, okay, more than one polypeptide chain, it is quaternary structure. Okay, quaternary structure. Primary, secondary, tertiary, it involves only one polypeptide chain. Okay, tapi kalau quaternary, 2 atau lebih. Okay, 2 or more. Okay, 2 or more. Okay, so name one example of fibrous protein. Kalau dalam diagram ni, collagen lah. Collagen, keratin, kan. So those are example of fibrous protein. And then figure 14 shows the structure of hemoglobin. So hemoglobin is a type of globular protein. Okay. So it is folded into specific shape. So for example, okay, for hemoglobin you have to know it is quaternary structure. Why? Because it consists of 4 polypeptide chains. Okay, 2 alpha and 2 beta. Okay, 2 alpha and 2 beta chains. Okay, so the question asks you to describe the structure of a hemoglobin. So 3 markah. Tadi apa? It has quaternary structure, 1 point. It consists of 2 or more polypeptide chains. So it is a conjugated protein. Why is it conjugated protein? Because it has a hem group on in each subunit here. It has a hem group. Okay, a hem group where inside it has iron. The function of iron is to bind with oxygen molecule. So for one hemoglobin, it can bind. One molecule of hemoglobin, it can bind to 4 oxygen molecule. Okay, so hemoglobin is the pigment inside red blood cells. Kan, it is a pigment inside blood cells. So inside a red blood cell. So hemoglobin inside red blood cells, there's abundance of hemoglobin. So if I'm not mistaken, you can check. There's about 250,000 hemoglobin pigments inside one red blood cells. Okay, so satu red blood cells maksudnya ada banyak okay, oxygen. Okay, it can carry so many oxygen molecule. Okay, last question for question number 2. I. So this is I. So casein is a globular protein derived from milk. Give another example of globular protein and describe the structure. Awak bolehlah cerita balik pasal hemoglobin ataupun awak boleh cerita pasal example enzyme ke hormon ke whatever. Kan? So globular protein asalkan awak cerita dia apa? Awak bagi soalan ni 3 markah kan. So satu markah awak bagi contoh hemoglobin ke, antibody ke, enzyme ke, hormones ke, lipoprotein ke, glycoprotein, nucleoprotein. Satu example, maksudnya satu markah lah. Bukan awak bagi 3 example, 3 markah. Satu example, awak bagilah banyak markah, banyak mana example pun, it is still 1 mark. Okay. So 1 mark for example. The second and the third mark is for your description. Okay, describe the structure. So description, it involves what? Because it is globular protein, or mesti cerita at least dia ada tertiary structure. Okay, so tertiary structure involves what? The polypeptide chain to coiled and folded. Okay, the polypeptide chain is coiled and folded. Okay, coiled and folded. Okay, so so this is to attain the tertiary structure. Okay, so the coiled and folded structure is due to what? Due to the interaction between side chains involving hydrogen bonds and hydrogen bonds. Tadilah ionic bonds, hydrophobic interaction, disulfide linkage, van der Waals interactions. Ada ionic bonds, ada 5 sebenarnya. Okay, so the interaction between the side chain ada 5 interaction. Saya repeat ada hydrogen bond, ionic bonds, hydrophobic interactions, disulfide bridge, and also van der Waals interactions. Okay, ada 5. Okay, so the coiled and folded is due to these 5 different types of interaction between the side chain. And then next boleh cerita lah tadi kan yang dia ada composed of tertiary and quaternary structure. Okay, tertiary and quaternary structure. And then some are conjugated protein. Kalau cerita dia adalah hemoglobin, awak boleh cerita lah it is a conjugated protein. Okay, it is a conjugated protein, it has hem group. Kalau cerita pasal hemoglobin, alright. And then because it is globular protein, you can add some more. It is soluble in water. Okay, soluble in water. And the last one, ada dalam nota awak, it has irregular amino acid sequence. Okay. Kita break sekejap for 5 minutes. Ya, you all boleh lah pergi toilet ke minum ke apa benda. Kita break sekejap. Tak sampai 5 minit pun. A few minutes. Okay. You all mana yang tak buat lagi entrance survey tolong buat Due date dia 20 hari bulan hari Ahad. Siapa online guna phone? Cuba angkat tangan.
Saya, madam.
So, tak panas ke phone?
Panas juga, madam. Tapi tak apa, boleh go on lagi.
Oh ya. Yeah. Dalam kampus ke? Dalam kampus ke kat rumah?
Dekat kampus.
Kampus, okay. Tak apalah, kita continue okay. Okay, so let's continue. Okay. Next question. Figure 15 shows structure of glucose and fructose that are isomers to each other. Identify and describe the type of isomer for both molecules. So glucose and fructose, we know that they are structural isomers. And for 1 point, structural isomers. And then second point, describe dah sikit kan, describe kenapa dia structural isomers, because they have the same molecular formula but different arrangement of atoms in the molecule. Different arrangement of atoms, kita tengok carbonyl group dia terletak berbeza kan. So kalau glucose, carbonyl at carbon number 1, fructose at carbon number 2. So glucose is aldehyde, aldehyde, ini adalah ketone lah. So ini aldose sugar, ketone sugar. Okay. So name the disaccharide form when glucose molecule and fructose molecule bind. Senang je, glucose and fructose adalah sucrose. The answer will be sucrose. And then carbohydrate in figure 16 is formed from 2 hexosugars. Kita ada kat sini adalah alpha glucose, ini adalah fructose lah kan. So alpha and fructose. So basically ini adalah structure sucrose. Okay, so this is sucrose structure. So name one function of the carbohydrate shown. Dia boleh bertindak sebagai energy storage. Okay, energy storage or source of fuel. Okay, fuel for cellular respiration, okay, to generate energy. Okay, fuel for cellular respiration to generate energy. Okay, so next question, figure 17 shows the structural formula of one organic molecule. In this case, it gives you the structure of alpha glucose jugalah, kan? So name the molecule and its macromolecule. Name the molecule. Satu markah alpha glucose. Markah yang kedua, macromolecule dia apa? Apa karbohidrat yang monomer dia alpha glucose? Kita boleh ada starch, okay, starch, glycogen, okay, starch, glycogen, carbohydrate even boleh. Macromolecule obviously carbohydrate, starch, glycogen is also accepted. Okay, so state another form of the molecule in figure 17 and differentiate its structure. State another form. Another form is beta glucose lah. Kan, beta glucose. Okay, and differentiate its structure. Apa beza dia? Beza dia dekat carbon number 1 ni, OH ke atas. Kan? OH ke atas. So, the difference is in the position of the hydroxy group, where the hydroxy group is located above the plane of carbon number 1. Okay. So next question. Okay, soalan ni kena tengok dengan teliti lah. So it says here F is a disaccharide, G is a polysaccharide made up of only molecule in figure 17. So that's why kan saya cakap bila dia bagi gambar rajah, awak kena refer sebab dia adalah information untuk guna untuk jawab soalan. Okay, so identify molecule F and G. So basically the monomer is alpha glucose. So apa yang alpha glucose jadi disaccharide. So F adalah maltose. Okay. Apa molekul or polysaccharides yang dia punya monomer dia adalah alpha glucose? Starch. Starch. Okay, starch ataupun glycogen ataupun amylose ataupun amylopectin. Okay. So D, figure 18 shows the formation of a disaccharide molecule. So which is J. So kita ada alpha glucose juga. So alpha dia akan jadi maltose lah kat sini. J is maltose. So join lah. So in the formation of the maltose sugar, so dia akan remove OH, dia akan remove H, and you can form oxygen in between carbon number 1 and carbon number 4. So carbon number 1 and number 4. So therefore the name of this bond is alpha 14. Alpha 14 glycosidic linkage ataupun alpha 14 glycosidic bond. Dua markah maksudnya apa? You draw the structure of the maltose correctly with oxygen in between. Satu markah. Okay. Second mark is you label the bond. Okay, alpha 14 glycosidic bond. So ini saya bagi kat sini lah gambar rajah. Saya contoh kat sini je lah kan. So your answer should be drawn here correctly. Okay, with all the atoms positioned correctly. Okay. So describe how the Benedict test can be determined. Can determine molecule J as reducing sugar. So kita tahu maltose ni is a reducing sugar. Why is maltose a reducing sugar? Because it has a free carbonyl group. Okay, a free maksudnya carbonyl group dia exposed to the reaction. Okay, so in order for you to detect whether your samples contain reducing sugar or not, you test with Benedict test. So macam mana nak tahu dia ada reducing sugar? First of all, you observe the change of color of the solution, the mixture between your sample that contains reducing sugar and the Benedict test. Benedict reagent. Observe, the first one, observe the change of color of the solution. Okay. So second point, kalau apa yang awak nak kena observe tu perubahan color kan. So the color of the solution will change from clear blue, okay, clear blue to a range of color ranging from green, yellow, orange, brown, brick red with precipitate. Okay, so the difference in color indicating different concentration of reducing sugar. So maksudnya kalau dia berubah daripada biru, clear blue to green, okay, the amount of reducing sugar will be low. Compared to if dia change kepada brown, the amount of reducing sugar will be high concentration of reducing sugar. Okay, and then the next point, kenapa boleh berlaku reaction perubahan color ni? Disebabkan oleh, awak cerita sikitlah reaction yang berlaku tu tadi. Because it has a free carbonyl group or a free aldehyde group, which is it involves in the reaction to reduce copper 2 ion to copper 1 ion. Benedict reagent ni komponen dia ada apa? Dia adalah copper 2 sulfate kan, SuSO4 kan. Okay, so dalam bentuk ion dia Cu2 plus, SO4 2 negative. So this carbonyl group is free to involve in the oxidation reaction. Okay, redox reaction basically. Redox reaction which is to reduce copper 2 to copper 1. Sorry, copper 2 to copper 1 kan. Copper 1. Okay, copper 1. So basically dia akan form daripada copper sulfate kepada copper oxide. Okay. Copper oxide itulah yang brick red precipitate. Right? So cerita sikit ni. Reaction yang berlaku ni. Okay. Free aldehyde group able to reduce copper 2 ion to copper 1 forming copper oxide. Okay. Figure 19 shows the molecular structure of a disaccharide which is malt sugar. Malt sugar, maltose lah. Kan? So this is basically the structure of a maltose. Is it a reducing sugar? Yes. Because why? It has a free carbonyl group. Okay. So dua markah. Jawapan awak yes. Yes. Is it? Yes. Okay. Ceritalah kenapa. Why? So because it has a free carbonyl group. Okay, a free carbonyl group. Aldehyde group which changes, and then another point, it changes the color of Benedict solution from blue to brick red ataupun tadilah color yang saya sebut tadi, green, yellow, orange, so sampailah brick red. Okay, and then awak boleh cerita juga it reduces copper 2 to copper 1 ion, the same answer as the previous question. Okay, suggest what happen when the sugar is hydrolyzed. Hydrolyze apa? Proses hydrolysis lah kan. Hydro involve water, to break bonds. Okay, so it breaks the bond lah kan, to form two glucose molecule kan. So produces two glucose molecules due to the broken glycosidic linkage. So dia satu markah je. Cerita glycosidic bonds is broken, so dapat markah. Produces two glucose molecule pun boleh dapat markah. So whichever, sama je. Ini sebab soalan ni satu markah. Kalau soalan disuruh dua markah, explain lah lebih macam ni kan. So next question, figure 20 shows the structure of a disaccharide. So tengok kat sini, ada galactose kan, kat sini OH dekat bawah. So ini glucose, alpha glucose. So name the disaccharide, it is lactose. Okay, lactose mestilah ada punya disaccharide lactose. Okay, so determine whether it is a reducing sugar or not. So nampak dia ada free carbonyl group kan. So determine whether it is a reducing sugar. Yes, it is a reducing sugar. Okay. And then next question, name structure M and label and the bond label N. M alpha glucose, label the bond between galactose and glucose, carbon number 1 kan, carbon number 4. So 1,4 glycosidic bond. Okay. So state the importance and the source of the disaccharide. Importance as source of energy, source milk. Okay, awak lapar-lapar minum susu kenyang, source of energy. Okay. So figure 21 shows the structure of a storage polysaccharide molecule in animal. Name the molecule in animal. So storage carbohydrate is glycogen. Okay, glycogen. Next, name the monomer in the molecule. Monomer dia alpha glucose. Name the bond label P, carbon number 1 and carbon number 6. So 1,6 glycosidic bonds. Okay. State how this molecule is different from amylopectin. Amylopectin, okay, the molecule is inside plant cell. Tapi kalau glycogen ni dalam animal kan. So bila dalam animal, obviously animal requires more energy compared to plant. Plant adalah organism yang static, duduk kat situ je kan. So obviously the storage carbohydrate for animal, the glycogen, the molecule will be much more larger. Kenapa dia large? Sebab dia ada lebih banyak branch. Okay, so glycogen will have more branch compared to amylopectin. By having more branch, meaning that it has more glucose, kan? So glycogen molecule is much more larger compared to amylopectin. First point, the molecule is larger. Glycogen is larger than amylopectin. Second, why? Because it has more branch compared to amylopectin. Okay, more branch. Okay, so figure 22 shows the structure of cellulose. Kan, so cellulose dia punya monomer dia adalah beta kan, OH dia kat atas, beta glucose. Draw one monomer of the above diagram. Draw lah structure beta glucose, ambil satu ni and then tambahlah OH dekat carbon number 1 kan. Okay, so that is the structure of beta glucose. So next, amylose and amylopectin are two components of starch. Describe the structure of amylopectin. Describe the structure of amylopectin. Apa dia? Amylopectin, apa monomer dia? Paling basic monomer dia adalah alpha glucose. The monomer for amylopectin is alpha glucose. 1 point. Okay. So amylopectin, does it have branch? Yes, it has. So cakaplah amylopectin has branches. Kan? It is a branched polymer. Kan? Second point. And then another point, awak boleh cerita the branches are held by 1,6 kan, 1,6 glycosidic bonds. The branches in the amylopectin is due to the formation of 1,6 glycosidic bonds. And then another point you may add, the monomers are bonded together through 1,4 glycosidic bonds pun boleh juga. Okay, the monomers, the alpha glucose are bonded by 1,4 glycosidic bond. Okay, next, compare the structure of amylose and glycogen. Okay, so amylose and glycogen. Kalau soalan dia tanya compare, maksudnya kena apa? Kena cari similarity, kena bagi similarity dan perbezaan. Persamaan dan perbezaan. That is comparing. Okay. So what is the similarity between amylose and amylopectin? The monomer are alpha glucose. Okay, the monomer are alpha glucose ataupun they both contain glycosidic bonds. Okay, they both contain glycosidic bonds, and then the difference. Okay, soalan ni you have to read carefully the question. You have to compare between amylose and glycogen. Comparing must be based on structure. Okay, must be based on structure. So kalau perbezaan ni cerita amylose are found in plant, glycogen are found in animal, salah. Okay, so you have to compare in terms of structure. So for amylose, it is unbranched linear. It is linear unbranched polysaccharides. Okay, so tapi kalau glycogen, it is highly branched. Okay, it is highly branched. That is structure kan. And then another point you can add adalah inside amylose there is only 1,4 glycosidic bonds between the glucose, and then tapi kalau glycogen ada 1,4 dan juga 1,6. Kan, 1,6 but dia ada branch. Okay. Okay, so boleh bertahan lagi ke?
Boleh.
Saya pun nak cakap, you all lah bagi jawapan pula, boleh tak? Boleh? Untuk soalan 4 dan seterusnya. Imran.
Sekejap, madam.
Ha? So kalau kita online ni memang susah lah saya nak serang you all kan. Dalam kelas nanti, tengoklah. Okay. Kita sambung. Saya nak kita boleh discuss as much as possible. Okay. Saya harap awak dah buat latihan ni kan. So bila awak dah buat latihan ni, you just mark your answers kan. So kalau mana yang salah tu betulkan according to the answer given. Kalau awak rasa jawapan awak salah, tapi awak nak defend betul jawapan awak tu jadi betul, awak bagilah tahu kan kenapa jawapan saya ni salah. Okay. Okay. Okay, next question. Figure 23 shows the formation of a type of fat molecule from K and L. Okay, so ni molekul K and L. M ni adalah fat molecule produced lah kan. So L ni adalah apa dia? Glycerol kan. Sebab apa? Dia ada 3 carbon. Sorry. This is not glycerol. This one is glycerol. K is glycerol. Sebab dia ada 3 carbon bonded to a hydroxyl. So L adalah fatty acids. Mana you all tahu? Sebab dia ada carboxyl group here. Okay. So K is glycerol, L is fatty acid. So draw the chemical structure of M. Join lah kan. So kalau in the formation of the triglyceride, M is the triglyceride. It removes hydrogen from the hydroxyl and OH from the carboxyl. So it releases each one a water molecule. Kan? So 3 water molecule will be discarded from the reaction. So produk dia adalah antara apa? Bond tu antara O, C dengan double bond O. Kan? So draw lah. Ada orang punya CHO, C double bond O, R. Kita punya side chain dia. So repeat lah 3 kali kan. Okay. So the bond between the glycerol and the fatty acid here is called as ester bond.
Which?
Dia ada soalan?
Tak tahu. Tak ada.
Okay. So next, name the bond that links the molecule adalah ester bond. Okay, ester bond ataupun ester linkage. What is the process involved in the formation of the glyceride? Esterification, accepted. Dehydration pun boleh, condensation pun boleh. Okay, sama je sebab dia apa? Remove water molecule. Okay, and then describe the solubility of M and the product of its hydrolysis. Okay, soalan ni dua benda kena describe. Satu, solubility of M. Is it soluble? No, it is not soluble. Okay, it is not soluble in water. M is not soluble. M ataupun triglyceride is not soluble in water. Okay, the product of its hydrolysis. Kita dah hasilkan triglyceride ni kan? Pecahkanlah balik jadi dia punya komponen. Komponen dia adalah glycerol and also fatty acid. So kat sini awak kena cerita. Okay, and the product of its hydrolysis. So boleh cerita, okay, produk dia dihasilkan glycerol kan, glycerol. So glycerol dia ada OH. So glycerol, is it soluble? It is soluble in water. So glycerol soluble, and then the other one is fatty acid. So fatty acid, is it soluble? No, it is not soluble. M not soluble. Okay, M not soluble, soluble. Fatty acid not soluble. Okay.
Madam, kalau ni ada wrong glyceride tak?
Ada, bio ada wrong glyceride. Dia kalau awak salah.
Kalau
Faham
saya
tak?
jawab yang have little or no affinity of water tu, salah ke? Ke betul?
Awak cakap have little no affinity of water, referring kepada apa?
M.
Dalam ayat awak tu memang cakap M.
Have little or no affinity of water.
Okay.
M.
Dalam ayat tu mesti cakap kan, M have little or no affinity for water, boleh diterima. Okay, little or no affinity tu boleh lah diterima. Okay. Sebab dalam nota pun memang cakap macam tu kan? Boleh. Okay, sebab dah ada dalam nota, okay, it is accepted. Okay, so next, different R groups in L. L apa tadi? L fatty acid, okay? Fatty acid produces different types of fat. Differentiate between saturated and also unsaturated fats. Okay, so saturated fat solid at room temperature. Unsaturated fat is liquid at room temperature. Satu perbezaan. Sebab ini dicakap secara general. So awak bolehlah bagi kan apa-apa perbezaan. So kalau saturated fat in animal, it is animal fat kan. Kalau saturated fats, it is fish, daripada fish oil ataupun plant. Okay, fish and also plant are usually unsaturated. And then kalau saturated fat, it has higher melting point. Saturated fat ni tak ada dalam nota, just additional. It has a higher melting point. Kalau unsaturated fat, it has a lower melting point. Okay. So next, name the process of converting unsaturated fat to saturated. It is hydrogenation. Okay, so unsaturated ada C double bond C kan, ada double bond kan. So jadi saturated, you break the double bond to attach more hydrogen. So the process is hydrogenation. Okay. Ini is of nak convert vegetable oil into margarine lah contohnya. Ada soalan?
Yang nombor 4 tu apa-apa jawapan general pun boleh
Ni.
4 atas.
Ni. Apa jawapan awak ada apa lagi?
Cakap pasal bond dengan number of hydrogen.
Okey, yang itu kan tak boleh diterima sebab number of double bond single Yang itu kan sebenarnya awak bezakan fatty acid. Yang ini di peringkat fatty acid punya perbezaan. Faham tak? Yang ini kan saya dah cerita kan. Kalau soalan dia cakap pasal fat, it is referring to the whole fat tu. This one is komponen dia, komponen dalam fat tu. Boleh?
Okey. Yang number of hydrogen pun tak boleh
Tak boleh sebab itu dah itu kat peringkat fatty acid. Okay, so figure 24 shows the formation of triglyceride. Complete the chemical reaction by drawing the molecular structure in the box provided. Ini awak kena draw fatty acid lah. Yang awak pecahkan. Yang pecahkan tambah H kat sini. Kan? So jadilah fatty acid 3. Kira 3 3. Baru dapat satu markah. Tulis satu je tak dapat. 3 all threes. Fatty acid you must draw. Okay, next, suggest 2 reason why aquatic animals in cool regions store lipid instead of carbohydrates. So lipid, kalau kita tengok kan haiwan yang duduk dekat tempat-tempat yang ada iceberg semua tu kan. So macam whale ke, seal ke kan. So dia gemuk-gemuk kat situ. So they are full of fats kan. So fat, kenapa? Fat is less dense than water. First point, okay. Fat is less dense in water. Ataupun for the same point, carbohydrate is denser than water. Carbohydrate ni dia lebih berat berbanding air molekul dia. Tapi lipid dia lebih ringan, okay, less dense. So and then second, by having fats, it provide buoyancy to the animal. Okay, it provides buoyancy. Sorry, dia boleh buat animal tu terapung lah kan, buoyancy. Okay. So and then yang ketiga kan, kat tempat yang sejuk mestilah dia nak perlukan lebih banyak tenaga, right? So lipid produces more energy, okay, compared to carbohydrates. Okay, it is it provides more energy lah kalau for lipid. And then another one, because they live in really cold temperature, really cold climate area. So it is useful for insulation, heat insulator, okay, insulation. Okay, so next figure 25 shows a lipid molecule. Identify this lipid unsaturated. Okay, jom. Identify this lipid. Okay, soalan ni dia punya jawapan dia kan. Kalau kita tengok kat nota you all, kita ada 3 class of lipid. Kita ada, kalau dia cakap 3 classes of lipid, so maksudnya this one, by you looking at this, your answer should be triglyceride. Okay, triglyceride ataupun fats. Kalau awak jawab unsaturated fats, boleh diterima juga lagi. Tapi tiba-tiba awak jawab saturated fats, terus jadi salah. Okay, unsaturated fat terus jadi salah sebab dia ada double bond. Okey lagi awak jawab fat, boleh diterima. Tapi tiba-tiba cakap unsaturated fat, dia jadi salah. Sebab ini soalan dia tanya pada peringkat lipid. Okey, lipid. Unless soalan dia ditanya, identify the type of triglyceride. Baru awak boleh cakap unsaturated or saturated. Okey. So draw a circle and name the bond formed in the lipid above. So draw a circle ni lah. So the bond is between glycerol and fatty acid. So with the bond must be between OCO ni, kan, OCO. So that is the ester bond. Okay, name the bond ester bond. Awak circle kan, lepas satu markah. Kan? So it is ester bond. Kalau terlajak sikit macam ni, jadi salah. Okay. So why is butter solid while oil is liquid at room temperature? Ni awak kena cerita lah dari segi dia punya komponen. So butter is saturated fats, satu markah. Okay. Butter is saturated fats. It is made up of saturated fatty acids, okay, with maximum number of hydrogen atoms. It has no double bond. Okay, it has no double bond between the carbon, and so therefore the molecule is not bent. Okay, the molecule is not bent, or it has no kink. Kink ataupun bent. No kink ataupun it is not bent. Okay, so kalau enough untuk soalan ni, enough awak describe salah satu saja. So kalau dah describe untuk butter, okey lah. Ataupun boleh describe untuk oil pun boleh. So which one? So kalau if you describe oil, explain lah oil. Okay, so oil is unsaturated fats. It is made up of unsaturated fatty acid. It has more than one double bond. It can accept one or more hydrogen atoms. It has kink ataupun it has a bent structure. Okay, it is a bent molecule. Okay, so explain how hydrogenation changes the properties of vegetable oil. Okay, so hydrogenation, it converts unsaturated fats to saturated fats. First point, converts unsaturated fats to saturated fats. And then second point, macam mana cara tu berlaku? By adding hydrogen atoms. Okay, and then another point you can add adalah hydrogenation changes cis double bond to trans double bond, okay, making the fat molecule a straight molecule, right? So therefore it is solid at room temperature. Kan? So cis kepada trans, fat molecule tu akan lebih straight. Okay, hydrogenation daripada bent dia jadi straight. Kan? So by having straight molecules, the fat molecule can stack upon each other. It can be closely packed together to form solid at room temperature. So hydrogenation is a process that is not good for your health. If you consume the type of food that has margarine ataupun trans fats, because they can contribute more to atherosclerosis, okay, blockage of blood vessels lah, kan, formation of plaque inside blood vessels. Okay. Okay, so next question 26 shows nutritional facts of for an unknown brand of fresh milk. Ini boleh contoh, ini project lah kot kan. So milk contains saturated and also unsaturated fats. Differentiate the structure of fatty acids. Soalan ni ditanya fatty acid, baru boleh cerita yang macam tadi. Okay, macam yang tadi awak tanya, baru boleh fatty acid. So unsaturated fatty acid have the maximum number, saturated fatty acid have the maximum number of hydrogen atoms. Maximum number of hydrogen atoms bonded to the carbons. Kalau yang ini able to accept, able to accept hydrogen atoms. Okay, able to accept one or more hydrogen atoms. Second, kalau ini no double bond, ini ada at least one double bond. Okay, no double bond, ini ada at least one double bond. Due to the presence of double bond, it makes the molecule yang ini bent. Okay, bent. Yang ini straight molecule. Okay, straight ataupun linear molecule. So example of fatty acid, cerita yang dalam nota. Kalau saturated fatty acid, you can have stearic acid ataupun palmitic acid in your notes, okay. And then kalau unsaturated fatty acid is oleic acid and then linoleic acid. Kalau soalan ni awak jawab butter, salah. Ni awak jawab fish oil, salah. Sebab fatty acid. So read the question carefully. So name the process to produce trans fat and give one disease. Okay, proses hydrogenation. Okay, hydrogenation satu markah. Disease tadilah atherosclerosis, cardiovascular disease, obesity, heart attack, stroke, kan, high blood pressure. So all those Diseases are contributed due to consumption of food that has trans fat. Okay. And then next question, figure 27 shows the structure of lipid. Kita habiskan soalan fat ni sikit lagi. Lidah saya dah naik 6 dah ni, duk bercakap je. Okay, figure 27 shows the structure of a lipid. In figure 27 shows, so identify glycerol and draw a box around it. Inilah glycerol kan? Glycerol. Satu markah dapat. Identify the type of lipid. Ada band kan? So lipid dia apa dia? Oh tak, ini ada ni. Sorry. Dia ada phosphate and choline. Ini apa?
Unsaturated fat.
Awak dah tipu macam saya ni. Sebab awak tengok ni kan. Tapi kat ni ada phosphate and also choline. So dia adalah phospholipid. Okay, it is a phospholipid molecule. Jangan tertipu ni so dia ada ni. Phosphate bonded to a choline. It has negative and positive charge. It forms the hydrophilic head. Hydrophilic heads. Kan? So hydrophilic heads. So this is phospholipid. Okay. So differentiate fatty acid K and L. K, ini baru ni. Fatty acid yang ni adalah unsaturated, ini saturated. So you have to differentiate between saturated and unsaturated macam soalan tadi ni. Okay. Cuma saya nak emphasize. Soalan ni awak kena tengok soalan dia nak perbezaan dari segi structure. Mesti structure perbezaan dari segi structure saja. Soalan ni secara generalnya. Dia tak spesifik kepada structure. So jawapan yang atas ni awak boleh apply. Kan jawapan ni maximum number of hydrogen atom, can accept one or more hydrogen bond, doesn't have any double bond, has double bond, kan straight molecule, band molecule. Sama je untuk jawapan ni. Cumanya cumanya untuk soalan ni dia lebih lenient. Kita tengok. Ini dia accept jawapan solid at room temperature. Any liquid at room temperature is also accepted. It is the component of fats that is solid at room temperature. It is the component of fat that is liquid at room temperature. Macam itulah jawapan dia. Okay?
Kalau cerita pasal kenapa dia solid dengan kenapa dia jadi cair boleh ke ada? Macam cerita dia linear linear, maksudnya kata yang fatty acid K dia band ke, dia ada king ke tak boleh?
Boleh cuma sama je. Okay. Band straight boleh terima. Okay, sama. Soalan ni lebih kurang sama macam tadi. Okay. So apa yang jawapan tadi betul, yang ni pun betul juga. Okay. So and then next question, explain how epiphytic properties of phospholipid contribute to the structure of cell. Okay, yang ni kita tak belajar lagi kot. Ada kita belajar tak istilah ni? Ini istilah ni sebenarnya dalam chapter 3. Epiphytic is a molecule having both hydrophobic region and hydrophilic region. That is epiphytic. Kalau phospholipid kan dia ada hydrophilic heads, hydrophobic tail kan? Itu epiphytic. Okay, epiphytic is a molecule having both hydrophobic and hydrophilic regions. Sama juga dengan protein embedded in the membranes, in the plasma membrane kan. This protein also have hydrophilic region and hydrophobic region. So that is amphiphytic. Amphiphytic awak jangan confusekan dia dengan amphoteric. Amphoteric molecule tu boleh jadi base and acid. Base and also acid. Tapi kalau amphiphytic, hydrophilic and hydrophobic region, dua-dua dia ada. Okay. Okay, so explain how amphipathic properties of phospholipid contribute to the structure of membranes. Kan, sebab dia ada hydrophilic heads and also hydrophobic tails. They will assemble to form a phospholipid bilayer. Okay, assemble to form phospholipid bilayer. It forms phospholipid bilayer, okay, of the cell membrane, where boleh explain apa jadi. Kan hydrophobic tail faces the interior. The hydrophobic tails of the phospholipid molecule faces the interior, bahagian dalam membrane, ataupun the hydrophobic tail pointing towards the interior ataupun isolated from water, and the hydrophilic heads points into the extracellular fluid side and also the cytoplasmic side. Okay, so situlah kat situ the formation of phospholipid bilayer of membrane. Sebenarnya soalan ni lebih kepada chapter 3 sebenarnya, tapi okey je. Okay. So last question, complete the table below to show the differences between triglyceride and phospholipid. Structure, phospholipid consists of a glycerol and 2 fatty acids. Okay, a glycerol, 2 fatty acid and also a phosphate group. Okay, a phosphate group. So kalau yang triglyceride ni function dia sebagai apa? Energy storage, kan? Energy storage, thermal insulation, macam contoh apa? Organism that lives in really cold area, source of energy, pun boleh juga. Okay. So saya rasa kita stop dulu sampai sini. Okay, tinggal soalan 5. So soalan 5 kita akan sambung minggu depan hari Jumaat. Dan minggu depan saya lupa nak bagi tahu you all, kita akan ada quiz. Kan, kita akan ada quiz on chapter 1. So saya nak tanya, you all nak kita discuss soalan 5 tu nanti hari Jumaat ataupun nak habiskan je?
Hari apa madam? Quiz tu?
Quiz tu akan diberi masa tutorial.
Dan
Sebenarnya dan tutorial. Sebenarnya okey, untuk you all, untuk group yang saya ajar tutorial, saya sebenarnya dah setting dah quiz you all dalam YouFuture. Dah setting elok-elok dah. Tapi dia tak appear lagi sebab saya dah letak date yang mana you all boleh start jawab quiz tu. Date quiz tu available daripada hari Isnin pukul 12.30 hari sampai hari Jumaat. Saya setting pukul 7 tak silap. Petang lah. Petang. Ya, ambil pukul 5 lah. Mungkin sebab saya tak ingat pukul apa saya setting. Daripada hari Isnin sampai hari Jumaat petang pukul 5. Okay. So quiz tu ada kat dalam YouFuture. Quiz tu 10 soalan objektif. Okay, 10 soalan objektif. Diberi masa 20 minit. Walaupun quiz tu available daripada hari Isnin sampai hari Jumaat, tapi once you click to answer the questions, klik je, kena terus jawab. Okay, tak boleh nak try usah-usah soalan, tak boleh. Okay, klik je, kena terus jawab 20 minit. Tak sempat jawab, terputus internet ke, try nak usah ke, dah tak ada peluang kedua. Okay, so make sure you are ready to answer the question. Then only you start to answer. Saya propose lah kan supaya hari Jumaat masa tutorial tu kita habiskan soalan nombor 5 ni, and then the remaining 20 minutes tu kita boleh buat quiz kan. So tak payahlah you all jawab dulu kan, kita buat sama-sama masa tutorial session tu. Okay, boleh? Dia ada je dalam tu, cumanya kalau nak jawab dulu boleh lah, cumanya saya nak you all jawab sama-sama time tutorial. Sebab kalau apa-apa senang, kan terputus internet ke apa ke, saya boleh bagger kat situ, saya boleh adjust. Okay, boleh? So kita kita stop dulu lah kot. Saya dah penat nak bercakap. Okay, boleh?
Boleh ada.
So saya harap you all boleh digest, boleh ambil apa yang kita discuss hari ni. Tujuan saya guna apa nama? Teams ni sebab saya nak recording dia. Kalau Google Meet tak boleh nak record. So ini ada recording. So kalau recording tu saya boleh download. Saya akan cuba upload balik and then saya akan upload dalam saya punya YouTube channel. Saya ada YouTube channel. So nanti bolehlah ambil kat situ. YouTube channel saya ada materials untuk fizik juga. So kalau nak usah-usah kat situ pun boleh. Sebab apa? Bahan tu adalah daripada saya punya husband saya lah. Husband saya dia buat untuk fizik. You all punya fizik lah. Fizik. Fizik ada dalam tu. Sebab dia ajar fizik. Okay, so itulah dia. Okay, so nanti kalau saya boleh extract, saya upload. Kalau tak, sampai sini je lah. Okay. Okay, so kita stop dulu. So thank you.
Ada apa nama YouTube channel tu?
YouTube saya cari FS to you.
NS.
FS to you. Fuzairi Syakirin. Kalau kenal lah Fuzairi, so dia ajar fizik, kau boleh tengoklah bahan kat situ. Channel tu dah lama dah menyepi sebab saya tak upload apa-apa pun sejak face to face mengejar face to face situlah. Okay, so kalau ada soalan boleh tanya, kalau tak kita stop dulu. So terima kasih.
Saya ada.
Okay.
Thank you, madam.
Okay, sama.
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 Thank you, madam.
Okay, sama.